quite a bit. But before I do that, I do want to make sure to acknowledge the collaborators who have been involved with the work that I'll be talking about today right up front, including folks from my own lab who have been involved in data collection or in discussions, and folks from other institutions, including Jason Rohr, who's here today. And in particular, I'd like to thank the undergrads who have contributed to this work, either in terms of effort or blood, sweat, tears. All of the above have gone into the data that I'll show you. So they're a big part of what our lab does. One nice thing about the last here is that we've covered a lot of the ideas in terms of background. There's very little new things that I'll be throwing in today. And one of the things that I think a lot of us can agree on is that in the realm of disease ecology, one of the fundamental challenges is how we understand infection, how we understand disease in the context of multi-host and multi-pathogen communities. And to kind of echo things that Vanessa said earlier, that Dana's written about in past papers, a big part of this is how we start to connect within host processes to between or even among host processes, right? And that brings together concepts ranging from super spreaders to co-infection to dilution effect, all under the same umbrella, and allows us to talk about things within hosts like susceptibility, immunity, and pathology, but then link that to transmission, of course, one of those gold standards in disease ecology, population regulation, and then something I would argue that we're still not that good at in disease ecology, which is really making the link to actual community ecology and even ecosystem processes. Much more challenging to do. And I think as we've seen that ecoimmunology really plays this key role in helping to link between those two scales. And I'm going to switch gears a little bit and talk about interactions between frogs and macroparasites, which is the system that our lab's been using for a number of years to explore some of these questions. And not quite as grotesque as Vanessa's slide here, but I'll try to up my game a little bit later. But there's tremendous variation in life history for these parasites, life history for the host, variation in abundance. We can find tens of thousands of parasites inside single frogs, which really creates a nice system for exploring some of these ecological interactions. Our lab's been lucky enough to be part of a group called the Amphibian Parasite Observatory, which over about the last 10, 12 years has collected around 15,000 data points using standardized methods to necropsy amphibians and get all the data on macroparasites from fairly good representation of the United States. We're trying to push that to larger regions of North America, but that's given us a lot of background data to put this in context. And just to kind of remind you, we've been talking a lot about microparasites, but to remind you some things about macroparasites that I want to emphasize is that these parasites are nice because they kind of force us to start thinking about interactions among species and, in fact, among different habitat types, right? In this case, terrestrial as well as aquatic habitats are intimately linked through the movement of these parasites, okay? And I'm going to be focusing, especially today, on trematodes and inside this sort of aquatic realm. Just to kind of remind you, when we're dealing with trematodes, there's usually a mollusk, in this case freshwater snail, and they're producing these free-living parasitic stages which run into all kinds of threats as they try to move from one host to the next. They're subject to UV radiation, to contaminants, to predators, all kinds of things that can inhibit their ability to move. And then ultimately they end up making a choice about who to infect. And there's usually going to be a variety of species present, even in a single wetland. And those choices can have impacts for their infection success, for pathology, for all sorts of things. And one reason I like this system is because it's fairly tractable to manipulate experimentally, even at realistic scales, as I'll show you a little bit later. Another reason is that in theory, larval amphibians are supposed to have fairly simplistic immune systems, although Stephanie may tell us that it's not quite as simple as we maybe once thought. You'll see more about that later. So I'm going to focus in particular on one parasite that our group's done a lot of work with called Rivaroi ondatre. It's a trematode. And one reason it's gained notoriety is it tends to target the developing limb tissue of a tadpole. Okay, so the free-swimming cercaria will burrow into that developing limb, that can cause these animals to die, or if they manage to survive, they can develop these sort of horribly misshapen limbs. You can get multiple extra limbs, missing limbs, or just kind of a variety of bizarre things that are even hard for us to describe. And the way that this happens is because, again, these parasites are burrowing in from the outside. They're contacting the host, secreting proteolytic enzymes, and essentially burning their way into the tissue. So it's a nasty process that causes a lot of pathology as it happens. And the hypothesis, of course, is that these malformations may actually enhance transmission to bird hosts, which are the definitive hosts for this trematode. And over the years, in our field surveys, we've seen that there's a fairly strong functional connection between the presence and the abundance of this parasite and the corresponding frequency of malformations in an amphibian population. Right? I mean, this log transform, but the frequency of malformations in a population can be 50%, or in some cases even as high as 100%. So this is a large fraction of the emerging frogs that can be influenced by this phenomenon. And every year or so, excuse me, passing through here, but people will ask me if a frog with an extra leg is better at jumping. And if I have the data, the answer is no, they are not. In fact, they swim slow, they don't jump very far, they get exhausted, their ability to capture prey is poor. So all things that we thought were pretty obvious, but eventually you have to collect the data to show it anyway. And this means that they show lower survival based on mark release recapture within a year, and then not surprisingly, they're less likely to come back and reproduce as adults. So there's suggestions of population level effects, although that's something that's very hard to show absolutely, so it's an area that still needs further work, but we suspect that it's likely. And this tendency of Rivaroa to cause malformations has gained it some notoriety. The Animal Planet recently ranked it the #1 most extreme animal transformer. And even though I watched that episode, I still have no idea what that means. Is that because they cause malformations? Is it because they have a multi-host life cycle? My guess is that it was just an attempt to capitalize on Optimus Prime and the Transformer movies. But in the world of parasites, you kind of take whatever press you can get. So it seems fine. So that's just all kind of background. Over the last few years, I think we've learned a lot about what drives disease risk in individual frogs. We know about the role of things like exposure. We know about the role of stage dependency. We know about behavior. What we know a lot less about is what's determining variation among species. And it's a common phenomenon that you can come up to a pond, you can find one amphibian species showing a high level of malformations and a lot of infection to go along with that. And yet another species there at the same time, even emerging at a very similar time, shows few malformations and very, very little infection. So we've been trying to think about why this is, and of course we already talked about this in several of the earlier talks, including Felicia's, and one interesting idea is applying this pace of life continuum. And just to kind of recap that the idea is that we can arrange species on this life history axis with fast species on one end. And those are species that grow quickly, they reproduce early, and they tend to have smaller body sizes. And then on the other extreme, we've got slow-growing species that tend to delay reproduction, have larger body sizes, longer lifespans, and so on. And this may also relate to immunity characteristics. Fast species, because they're spending less time on Earth, right? They're less likely to get exposed to parasites. They'll probably be exposed to fewer parasites in terms of absolute number and in terms of diversity. Maybe therefore they invest less resources in defenses, and those defenses they have might be more likely to be non-specific and constituent. Slow species, on the other hand, they're going to be around, they're going to get hit with parasites, it's just a question of when. Maybe they're more likely to invest in costly defenses, especially those that are going to have a low rate of self-damage. And the simplicity of this framework gives it tremendous conceptual appeal, but of course it can be hard to apply empirically, as many of you know. And one reason for that is the challenge, especially if you're out there actually measuring infection in the field, of decoupling exposure from defenses. Right? Because exposure and defenses are both going to be influenced by life history. And if you have something like a fast species, and this is a chorus frog that I'll be talking about several times today, they develop very rapidly to metamorphosis. They only live a few years as adults, right? And here's a bullfrog on the right, much larger bodied species. They can take a year or more to metamorphose, and they can live 15 years as adults. And not surprisingly, there's big differences both in average infection and in parasite richness. But how much of this is due to defenses, and how much of this is due to exposure history? And the approach that we've been taking to try to decouple those two things is just taking an experimental approach and doing laboratory exposures to animals of different species that vary in this life history gradient. So we've used 13 amphibian host species, and we're exposing these species to this trematode, Rivaroion datre. And what we do is we bring egg masses into the laboratory, we allow those to hatch, and then we're exposing tadpoles to different dosages of the parasite. Okay? And we have to do different dosages again, because remember, this is a macroparasite, so pathology is intensity dependent. So we're actually trying to generate those pathology curves. And then the two main classes of responses we're interested in is, one is infection. So how many parasites do we get back of those that we add to the tadpoles? And then in terms of pathology, we're looking at several responses: survival, the likelihood these animals become malformed, and various measures of growth and time to metamorphosis and so on. Okay? And then we're interested in testing how different life history variables or life history axes help predict these responses. Oh yeah, this is what an experiment with one species looks like. And so each of these little lunch boxes has a tadpole in it, and then you're counting out specific numbers of parasites and adding them into these containers. Jason and Matt, if you were still here, could attest to the time-consuming nature of these studies, which is why we only have 13 species. It doesn't sound like a big number, but these things take forever to run, because you then have to raise these guys all the way up to metamorphosis. So 13 species, not a lot, but it's taken us a long time just to get that number. So I'll just focus on those 13 for today. And here's the data for those species. And I know these plots are kind of small, so I don't want you to focus on any of the particular species. The main thing I want you to see is just two general trends. Okay? This is parasite exposure on the x-axis. Remember, we're using multiple dosages for each species. And notice that survival, which is the black line in general, is decreasing, and oftentimes malformations are increasing. Right? So you get hit with more parasites, you're more likely to die. If you don't die, you're more likely to become malformed. But what's interesting is the variation among these species, and there's quite a bit of it. And again, what we wanted to do is try to relate that to some of those life history predictors. One thing that's slightly different for amphibians, although this actually can be true for a number of different groups, is that when you put all the life history variables into a principal components analysis, you don't necessarily just get one really clean life history axis out. Right? Here we're actually getting two. One we might consider the growth and development rate. Okay? Variables like size, maximum body size, size of maturity, time to metamorphosis, lifespan, all of those are loading positively on that growth and development axis. Okay? But most of our variables related to reproduction are loading on a second axis. We've got clutch size, range size, and egg size being an indicator of parental investment into the offspring. And those are all loading on the second axis. So this actually gives us kind of an interesting opportunity to put both of these variables in, both of these axes in, and explore their relative explanatory power. So here's those results, and these are the three plots just representing our main response variables again. This is infection, this is likelihood of dying, and this is likelihood of becoming malformed. And what we found is that only one of those axes came out as being an important predictor for all three response variables, and that was that growth axis. Okay? And that shows a negative relationship with all three responses, suggesting that these fast-paced species that are way over here, they're on the left side of the x-axis, they're less likely to support infection. They're more likely to support infection. They're more likely to die, and they're more likely to become malformed. Whereas these slow species over here on the right, they're getting less infected, and they're less likely to show pathology. And if you break this down a little further, if you start looking at the individual variables, the individual life history variables that are accounting for this trend, the main thing you notice is that the important ones relate to larval development for these amphibians. Things like size at metamorphosis and time to metamorphosis. Okay? And this makes a reasonable amount of sense because these parasites are being acquired during the aquatic phase of these animals' lives. Okay? So things like body size while you're a tadpole and how long you're a tadpole, both of those things are helping to explain these pathology responses. And my guess, and we're still working on this part, is that there's really two different things going on here. One is a very simple one, which relates to tolerance and body size. Right? If you're a tadpole of a fairly slow species of amphibians, a large-bodied species of amphibian, right, each of those parasites that's burrowing into you is causing less damage proportionally relative to your total volume. Right? Whereas if you're a larva of a very small-bodied amphibian species, every one of those things coming into you is disrupting more tissue proportionally of your body and more likely to cause you to die or become malformed. The second part is that we think there's variation in infection resistance at both short as well as longer timescales. This is newer results that we're still working on, working all the way through. But if it takes you longer to develop to metamorphosis, you have more time for parasite clearance. Okay? And these are data on bullfrog larvae that we all exposed at time 0, and then we're monitoring them over time, and there's this nice sort of decay of total parasites that are recovered from the animals when we necropsy them. We expect this is related to adaptive immunity, although we haven't actually measured any immune parameters to get into that yet, but there'll be some coming. So that's kind of a slower clearance-related process, but we also see variation in very short-term responses to infection that we found really intriguing. So I didn't focus on it in the earlier plots, but one species that actually stands out a little bit as an outlier is the gray tree frog. Okay? Somebody's excited out there. They do get infected with this parasite, but they seem to have an ability to wipe it out pretty quickly. And what I'm showing you in this slide, this is actually a tadpole that's been anesthetized. Its head's up here somewhere, and its tail's down here. And these are all parasites that have been fluorescently marked. And these ones here have all already made it inside the host. They've dropped their tail, and they're forming cyst stages. Okay? The limb itself is actually right here, if you can see it. So the parasites are showing that tight distribution right around the limb. These two haven't yet fully penetrated the host, which is why they still have their tails. That's what you're seeing there. Okay? So we do know that they get into the host. They manage to penetrate the host. They start forming cysts, but within about 24 to 48 hours, they'll be gone. There'll be no evidence that they were there anymore. And one of my students, Brian Lafont, has been playing with trying to understand what's going on here. And one of the simple first studies he's been doing is using corticosterone to try to knock out the tadpole's defenses. And there are data on this spot. I'm just hiding it for the moment to kind of explain what's actually going on. What he would do is, in the cord treatment, he was exposing tadpoles to cord for 2 weeks. Okay? And then after the 2 weeks, he would basically he would stop the cord treatment and then expose animals to these larval trematodes. And then he examined them, he necropsied some of them at 4 days, and then again, other animals at 14 days. And we also had a control treatment, which they weren't exposed to cort, but they were exposed to parasites. And then a vehicle control of a very, very low concentration of ethanol. We use ethanol to dissolve the cort into solution. Okay? And this first part of the plot is data for Rivaroa, and it's a little hard to see what's going on here because only one bar shows up. But interestingly, that's the animals that were treated with corticosteroids. So at 4 days post-exposure, you're getting no parasites back essentially in the control or the vehicle control, but this really strong increase in infection if animals were exposed to cort. But this effect is essentially gone 10 days later, so almost no evidence anymore. And again, this is 14 days after we stopped the cort treatment. A little hard to see on this plot, but we repeated this for 2 other trematode species. And here it's much easier to see what's going on. And in all cases, the animals treated with port have somewhere between 50% to twice as many parasites as the other treatments do, and this effect is weakening by 2 weeks or 10 days after the first examination, but you can still see it significant for these other two parasites. And Marty told me I'd be summarily kicked out if I didn't have any immune parameters in here. So, do eosinophils count? Okay, perfect, cool. So these are based on the blood smear data from these sort of same animals that I showed you in the previous slide. And what we think is going on is that circulating eosinophils, which of course play a role in attacking things like macroparasites, may be explaining this pattern, which is consistent with some other work that's been done on amphibians. And here you can see the animals treated with quartz show this strong depression. Here I've got data for percent eosinophils relative to 2,000 erythrocytes, but you can see the same thing if you look at absolute numbers as well. And by 10 days later, that effect, they've almost recovered up to the same level as the other treatments, but they're not quite there yet. Okay. Now, as a community ecologist, what I'm really interested in is we know there's this variation among species. We can predict some of it at least based on life history. But how does that influence among species transmission within real systems? And this is kind of building on, I was going to say that my talk is basically a mashup of Vanessa and Felicia's talk, so you'll see a lot of the same ideas coming in throughout this presentation. So coming back to this idea of the dilution effect, if we know that species vary in host competency, what happens when those species co-occur together? And one of the first studies we did just to try to explore this is taking two species, one that's very low susceptibility, that gray tree frog that I was just telling you about, and the other which has high susceptibility, which is the American toad. And if you maintain these species together, an interesting thing happens. And that is that gray tree frogs and toads raised together, there's actually fewer parasites total in that community than there are when there's toads basically isolated by themselves. Okay? And that's because some of the parasites are trying to infect the gray tree frog and they're subsequently dying as a result. And that's offering this really interesting indirect form of protection for the toads, because the toads are highly vulnerable to this parasite, and if the tree frog is sponging some of them up, you can get this strange form of parasite-mediated facilitation. Okay, so this at least tells us that who's in the community matters. This is a very simple community. We're only doing two species. I just sort of artificially put them together. So what we've really been trying to do in the last few years is expand this approach and going into realistic systems, looking at the assemblages of amphibians that are there and trying to understand how the actual observed assemblages that we see influence patterns of transmission for this pathogenic parasite. Okay? And these are, see, here we go, pond. We're in the pond room. This is appropriate. This is the kind of study system that we work in California. We sample a lot of ponds out there. And what we've been finding is really two interesting things that relate to the dilution effect. One, and again, this builds directly on the type of thing Felicia mentioned for the mammal community, is that larval amphibian communities show a very strong pattern of nestedness. And I'm showing a slightly different plot from what she showed that just kind of summarizes data from several hundred wetlands that we work in California. And it's this sort of triangular pattern that gives us evidence of nestedness, right? Such that low diversity communities are essentially a perfect subset of higher diversity communities. If these data were not nested, you'd get a lot more dots up in this area here. So they're nested, which is important, but what's even more important is the relationship between the order of assembly and essentially the competency of these different hosts for Rivaroia. And we have data at least for 4 of the amphibian species, and we can show this relationship between that the most competent species also tend to be the most common ones that we encounter in these ponds. Okay? So I'm missing data for Rana draytonii and Erythema californiensis. These are both federally endangered species, so we haven't been able to do experiments on them, but we're now using some of their congeners to try to fill in this gap so we can round out this plot. So armed with this kind of information, again, what's nice about these larval amphibian and parasite interactions is we can start to manipulate them in semi-realistic environments, and to do that we use these mesocosms. And we can essentially build our own pond communities, and we can vary density and we can vary species richness, modeling after what we've seen in the ponds that we've sampled in California. And this basically allows us to get at the question, how does the density and diversity of these amphibian assemblages influence the fraction of parasites that you get out relative to those that you put in, and things that we might care about like host fitness, right? Especially for amphibians, which are a declining group of vertebrates. And here I'd be remiss, I got to give a quick shout out to one of my grad students, Dan Preston, and he just played a Herculean role in helping to run this experiment. Because remember I told you that in order to do this sort of thing, we have to collect lots of snails. They all start releasing these free-swimming stages of parasites. We harvest those, we count them out, and then we add them to the mesocosms in the middle of the night, okay? And the reason we do that is because these parasites are only active usually around the hours of about 10 p.m. and 2 a.m., okay? So we didn't want to alter their biorhythms, so we just tried to leave things as they were. But it meant a lot of very, very late nights. This is Dan out with a headlamp adding parasites to each mesocosm. And we did this for about 10 days and added 5,000 parasites to each tank. So very, very labor-intensive experiment, but fortunately for Dan and for me, it did generate some really clear data, which is always nice. And these first are the results for host density. Okay, so this is just one species communities. Now we're just varying host density here along the x-axis, and there's an increase in parasite infection success. And then more interestingly, here's the data for species richness. So we go from 1 species to 4, following the patterns that we see in the field. And you can see there's this pretty sharp reduction in infection success. And one reason why that's impressive is if you actually look at this 4-species community, which has these bullfrogs in it, the amphibian biomass in that system is quite a bit higher. So if transmission were modeled solely on a biomass basis and we didn't think about species identity, we would actually expect there to be more infection success over here. And it does seem to matter for host fitness. We're still analyzing some of these data, but again, because Riboroia infection predicts malformations, and it can also reduce amphibian growth and body size. One thing we see for this kind of focal species, the chorus frog, is that in the richer community, they show larger body size and fewer malformations. Okay? And normally you might think more species here, there should be more competition, or at least interspecific competition, but we actually sort of see the opposite effects in terms of fitness.
So you did say, is it substituted design, or?
We did both substitutive and additive. Yeah, so, oops, let's see, go back here. So we actually did 3 different densities in the single species communities, and then did the 2 and the 4, and that way we could analyze it with either substitutive or additive design. All right, where are we?